Hi everyone! Today we're going to be talking about Foraminifera, aka Forams for short, but this is one of the last phyla we're going to be talking about for invertebrate paleontology. Now we see a bunch of different forms on this slide here, but what really are these forams? Well, forams are protists, aka they belong to the protist kingdom, which are under the eukaryote domain, so they are eukaryotes, however they're not technically animals because a protist is any eukaryotic organism that is not an animal, plant, or fungus, so they're technically not animals, but they're animal-like in that they're more like animals than they would be a plant or fungus. And in terms of the more specific categories than kingdom, they belong to the phylum Sarcomastigophora, they belong to the subphylum Sarcodina, and they belong to the class Granuloreticularia, and they belong to the subclass Rhizopoda, and they belong to the order Foraminifera. You will hear throughout this video me calling them either planktonic or benthic, and planktonic just means they live in the water column, they kind of float there, and then they die and fall to the bottom sediment. But benthic forams refer to those organisms that live on the seafloor or in the seafloor if they burrow, which they sometimes do just a little bit, but we'll get more into planktonic versus benthic forams when we talk more about their morphology and their ecology. But in terms of abundance and diversity, foram density can exceed 1 million specimens per square meter of sediment on the seafloor. And much of the seafloor above the CCD, because of this abundance of forams not only living on the seafloor but also falling down when they die from the water column, is made up of calcareous ooze, which is basically their calcareous skeletons falling down to the seafloor or dying there, and they end up causing this calcareous ooze that ends up making up a lot of that sediment as it gets buried, compacted, and preserved. And it's not only seen on the seafloor underneath the water, but even on beaches we can see a lot of the skeletons of these organisms, because sometimes beach sand may be completely made up of these foram skeletons. And in terms of diversity, forams are more diverse than any other groups of marine animals or plants. But what are forams useful for? Why do we study these things? Well, forams can be great for telling us about the past. Forams make great index fossils for something called biostratigraphy. If you want to date rocks or figure out the age of sedimentary rocks, it's really hard to do with isotopic dating methods because with sedimentary rocks, all the grains have come from different sources, and so you're going to get a lot of different ages. But if you have index fossils, you can use them to relatively date those rocks and then correlate those rocks through strata from all over the world. But these forams are not only great index fossils for figuring out what time we're at in the geologic timescale when we're looking at rocks, but also figuring out what temperature the ocean was at then, what chemistry was going on in the ocean back then, what depth in the water that sediment was deposited in, because forams have very specific temperature, water chemistry, and water depth needs. That means they're very useful for paleoclimate, paleooceanography, and paleobathymetry, respectively. And because of their very specific environmental needs, we can also easily reconstruct depositional environment using our knowledge of what forams are around in those strata, and that has been useful for finding oil. So what are these specific environmental needs that these forams have? Well, let's go over a little bit of foram ecology. We're going to talk more about this later in detail, but in general, forams can be sessile or immobile, but they are capable of moving up to 1 centimeter per hour. Okay, I know you might be thinking that that sounds really slow, but for a 1 millimeter size organism, that's pretty fast. Also, many of them creep along surfaces, but some benthic forams, meaning the ones that live on the seafloor, can burrow into the sediment as deep as 10 centimeters. However, planktonic forams, the ones that don't live on the seafloor, live in the water column and kind of sink up and down until they die and fall to the sediment. But this sinking up and down is very similar to cephalopods, which we talked about in the cephalopod video. And we talked about how some of them also do that in the water column, and that's because of the way they control gas in their chambers of their shells. And these planktonic forams are similar to that in that they probably sink up and down via gas control in their protoplasm, or their living material. And I'm going to bring back this figure later and we'll go over it in more detail. So if you're not sure what the heck's going on there, don't worry. We'll talk about that when we get into the more detailed ecology. But before we get into that, let's talk a little bit about foram reproduction. Benthic forams reproduce both sexually and asexually. They alternate between sexual and asexual forms as they alternate generations. The asexual or microspheric adults split their nuclei and cytoplasm into several parts. Then it expels them from their test, and the young are planktonic until they settle to the bottom sediment and grow a new test. And so we can see over here this asexual pathway to the sexual adult. But what does this microspheric and megalospheric terminology mean? So microspheric is a term used to describe the asexual adults in forams because they have a relatively tiny first sphere, which is like their first shell that they grow off of as they grow their test and their chambers. Kind of similar to what we talked about with the cephalopod shells and a lot of different mollusks as well. They have a first part to their shell and then they continue to grow off of that. In forams, that's typically called the proloculus, and in mollusks, sometimes we call that the protoconch, but it's this first part of the shell. And this sphere is micro in the asexual adults and megalo or relatively mega in terms of the sexual adults. So it's much larger in the sexual adults. And we can see that in the sexual adults or the megalospheric adults, we have the nuclei that will disintegrate and form millions of gametes, reproductive cells. And these gametes will collide in the water column with other gametes and form zygotes, which will grow into the asexual or microspheric adults. And so we can see this pathway of sexual reproduction in this figure as well. So what about the more specific ecology that I said we'd get into? So this is a figure that is just really helpful for remembering which forams live in different environments in shallow marine systems. So what we see in this figure is benthic forams on the left side and planktonics on the right side. And then in this little part in the middle, we have a marine environment going from lagoonal to near shore to outer shelf to upper shelf to abyssal plain. And we'll talk about which forams live in each one of these environments. But first we have marshes, which aren't necessarily shown on this figure, but we'll talk about them briefly. Basically, in marshes, you will get low diversity agglutinated forams. And then once you get to lagoons and estuaries, as shown here in green, you get perforate calcareous forams. And we'll talk about different composition of forams when we get to the morphology, but basically perforate just means they have pores in their test, and calcareous just means they're calcite forams, which isn't a common characteristic among all forams, and we'll get to that later. So anyway, they're perforate calcareous forams such as the genera Ammonia, Elphidium, and Rosalina, shown in these lagoonal images over here, respectively. And then we have near shore, shown in blue here on this environment figure, and then up here, the forams that are common in the near shore environment are shown. And these include lagoonal forms like we just talked about above, but also porcelaneous miliolines. So miliolines are forams that include genera such as Quincalocolina up here and Triloccolina. And these forms are called porcelaneous because they are made up of smooth calcareous material and look like porcelain. In the outer shelf, aka 50 to 200 meters in water depth, we have benthic forams shown up here, such as genera Cassiolina, shown down here in the upper shelf environment, but it's also present in the outer shelf environment. Then we also have Amphistygina, and that's shown over here in the outer shelf category. And then we have Nonian and Nonianella, which are both shown over here in the outer shelf category as well. And we also have planktonic forams show up in the water column over here. They aren't typically found, or they're more rare in the near shore and lagoonal and marsh environments. And so they show up in the outer shelf. And then as we'll see, they also show up as we go further offshore. Next, we have bathyal depths, or the upper shelf environment, aka 200 to 250 meters in depth. And we have in this environment benthic, biserial, and triserial forams. And we'll talk about what biserial and triserial means. You can kind of already see it in these forms like Bolivina, which is biserial, aka it grows a double layer of chambers, and then we have Foramina down here, which grows kind of a triple layer of chambers, and we'll talk about the morphology when we get to test morphology. Maybe I should have gone over that first, but we're just gonna go with it. So anyway, these like to live in this upper shelf environment, as well as Euvagerina and Cassidolina. We talked about Cassidolina, which also sometimes lives in the outer shelf environment, but Euvagerina is shown down here. We can see that's a uniserial benthic foram. And in the upper shelf, there's also imperforate porcelaneous millilines, such as the Pyrgo genus. And this is shown over here in this spherical-like form with this outer edging around it. And this is similar to the porcelaneous millilines in the nearshore environment, but instead of perforate, it is imperforate. And obviously it doesn't live in the nearshore environment. Then we have planktonics, also abundant in this upper shelf region from 200 to 250 meters. Then we move to the abyssal plain, and the abyssal plain above the CCD, which is the carbonate compensation depth, aka carbonate material like calcite and aragonite that many of these planktonic forams build their tests out of, dissolves below this depth and does not above this depth. So above this depth, or above the CCD, planktonics can be present and are abundant in the water column, but as you get below that depth, which is typically around 4,000 to 5,000 meters, you will only see agglutinated forams below that CCD. And that's because agglutinated forams, which we will talk about in 2 slides, I promise, are forams that are not made up of carbonate material. Instead, they are made up of glued together random particles like sand grains or foreign particles at the bottom of the seafloor that aren't dissolved as easily as carbonate material. So agglutinated forams past this depth are common. But what about test morphology? I threw out a bunch of terms at you like agglutinated and protoplasm and test and calcite and all these things, but what's it all mean? So forams are single-cellular organisms that have a shell, but we call it a test in terms of forams because who knows why, it's just what we do. And usually it's made up of calcite or aragonite like we talked about previously, but we also talked about some forms that aren't, so we'll get to that. And then they also have protoplasm, which is basically their living material in their cell, and then their cytoplasm is, like in any cell, the material that everything else is in. And so their protoplasm encompasses everything in the cytoplasm as well as the cytoplasm itself. So that's not very different than any other single cellular organism, or even multicellular organism for that matter. But in terms of forams specifically, they have pseudopodia, which obviously means fake feet, but they're kind of, in my opinion, somewhat real feet. I mean, these things shoot off the forams kind of like cilia, and they help them move and capture prey. And their prey that they're catching with these pseudopodia include things like diatoms, coccolithophores, radiolarias, silica flagellates, and even small crustaceans. And if you're not sure what some of those terms are, we're going to talk about all of these in some upcoming videos, so don't worry. But what about those other terms I used to describe forams in the ecology slide? Uniserial, biserial, triserial, all of these things. So let's go over what they mean. Sometimes forams can be unilocular, like we saw in the Pyrgo genus that lived in the upper shelf environment. And that just means that it has only one chamber. However, that's only common in a few forams, whereas many have multi-chambers. They have a lot of different chambers, but the way that they grow or add on these chambers is different. And that's what causes so much of the diversity observed in forams. So one way in which forams add on their chambers is in a single row, such as uniserial forams. It's just one single line of added on, increasing in size chambers that the foram has. Then you can also have biserial, which is 2 chambers per row that go up in a line similar to the uniserial. Then you have triserial, which is 3 chambers each row. Then we have coiled types, kind of like cephalopods, honestly, like microcephalopods that can coil in one plane, like planospiral cephalopods, similar to certain ammonites. Then you also have trochospiral forms that aren't really shown very well here in this image because we're looking at it from above and below and not from the side. So you can't see that the coil is actually not in one single plane. It's actually kind of like if you've ever seen a snail shell. The coil goes outward a little bit, and this outward coil is not one single plane, obviously, and therefore not planospiral like the planospiral one we see over here, and it's actually trochospiral. And the tippy top of the hat-looking spiral is the umbo, and the bottom part is the umbiculus. And then we have involute and evolute here. Involute just means that the chambers of the previous whorls are enveloped or covered up by later whorls. But in terms of evolute, that just means that the inner whorls are not enveloped by the later whorls, and so you can still see the initial whorls and initial chambers that have not been covered up by the later chamber growth things. And then we have the aperture, which is just the opening that's on the largest chamber or the most recently grown chamber of the foram. And in some forams, there are secondary apertures along the other chambers of the test. And the lines between the chambers are called sutures. And so we can see these sutures labeled in all of these forams shown here. And sometimes they like to get fancy with their sutures, and sometimes they have what's called septal bridges instead because they just wanted to be extra. And then we have more possible decorations down here like spines, ribs, turbicules, and keels. For example, we have spines shown on this guy over here. Then we have this guy over here with no spines, and this keel is this like outer edging part here, which we saw on some of the forms we looked at in the ecology slide. And then they have the other normal things like chambers and sutures between their chambers and a coil direction, which can sometimes tell us a little bit about temperature. And they have an aperture lip, an umbilical cavity, the proloculus, the first chamber. They also can have pores in their test, and we'll talk more about the test wall structure and composition on this slide. So we talked about agglutinated forams and calcareous forams in the ecology slide, but we also have some other terms on this slide. What are these? Well, we have agglutinated forams, which are kind of like random grains coming together to make their test. We have microgranular calcitic or calcareous forms like these Fusulinids shown here. Then we have porcelaneous forms like we saw in the miliolines, and we can see that these are also calcite, but they look a lot different than the microgranular forms, and that's just because they have a different crystal structure of calcite and they come out more smooth and shiny. Then we also have hyaline forms, which appear glassy because they're made of silica instead of calcite or random grains. The agglutinated forms, like we talked about earlier, are not typically made with calcite particles. Typically, they're like sand grains and other foreign particles, and they can live deeper than the CCD because they don't have calcite that will dissolve. They just have random grains that won't dissolve, and therefore they're fine and can live below that. However, calcitic forms cannot live deeper than the CCD, and they can be perforate or imperforate, so they can have pores or have no pores. And then the porcelaneous forms are typically smooth and shiny, like we see here, and have a high magnesium content, which might be the reason that their crystal structure is a bit different, causing them to look porcelaneous rather than microgranular. So what about foram classification? We already talked about how they are protists and they're in this phylum, subphylum, class, subclass, and order listed here that I don't want to repeat because they are very hard words. But in terms of suborders of forams, these include Allogramina, Textularina, Laganina or Laganina, Fusulinida or Fusulinina, Miliolina, Rhodolina or Rhodolina, and Globigerina. And the first one is characterized by single-chambered or unilocular, tubular, round, or flask-shaped forams with organic tests. The second includes agglutinated forms. The third is typically uniserial and sac-shaped. And the fourth, or the Fusulinids, which we'll talk about more in the next slide, are spindle-shaped and very abundant in the late Paleozoic. And this abundance in the late Paleozoic, as well as their very specific morphology and their geographic widespreadness, makes them really good index fossils. But we'll talk about those in the next slide. Then the miliolines are porcelaneous tests. We saw those in the ecology as well as the previous slide when we talked about different compositions of foram tests, and they're shiny calcareous tests that typically lack pores, aka imperforate. And Rhodolina includes multi-chambered forms with spiral or hyaline tests, so like glassy tests. That includes mostly benthic forms forams, and then Glabidrides are planktonic, not benthic, and they have bubble-like chambers that are arranged in expanding spiral. And I like these because they're typically easy to remember because Glabidride sounds kind of like globular, and they look globular because they're bubbly. But now I want to talk a little bit about Fusulinids because these are really important index fossils, and I don't know about you guys, but my professor really liked to focus on these, probably because he studied them for a lot of his life and he was really an expert in them. But anyway, these guys are like little rice-grained, football-shaped little things, and I'll show more cross-sections of them here in a second so you can see their more internal complex structure. But these guys lived from the Mississippian to the Permian, and in terms of their evolution, they evolved from small simple forms like Millerella, this tiny form down here that was relatively simple in internal structure, to much larger and complex forms in the late Pennsylvanian and Permian periods of the late Paleozoic. And this widespread domination of these forams led to them being able to be correlated across all continents, making them really great index fossils. And another thing I think is just cool is that they got as long as 4 inches. You can see from this size, around 0.2 millimeters to 4 inches. That was an incredible evolution and really, really crazy for something that's single cellular, guys. But in terms of their morphology, which can be really important if you are maybe taking a test on forams and your professor likes Fusulinids, I'm going to go over some of the terms that go along with Fusulinid morphology. So what we can see here is their football shape, and we can see this also in this rock slab that's in the background here. They have a football-like shape in their axial section, or their axial cut along the long ways of their body, and then they have a rounder shape if you cut them in the equatorial section, so along their like equator, or this like cut right here that you would do. If you cut that, it would be a circle. If you cut along this way, it would be a football shape, and that's what we're seeing in this rock slab. And so if we look at some of the labels here, we can see some similarities with the overall general foram morphological terms like proloculus, their first chamber, which can be relatively tiny or relatively large depending on whether they're microspheric or megalospheric at the time, aka an asexual adult or a sexual adult respectively. We also see some other terms listed here that are also labeled. We have the antitheca, which is the line that marks the outermost edge of the growing surface. Shown here, it's kind of ripply, and these are just folds, and sometimes these folds can cause an overall structure typically termed fluting or septal fluting. And then we also see chambers. That's another common characteristic among all forams we've already talked about. And so these are obviously multi-chambered, but they're not uniserial or biserial or any of those morphologies. They're actually coiled, and they are technically planospiral, but instead of coiling in one plane and then just being like a thin coil thing, they're planospiral, but they're also called fusiform in their morphology because they have this elongated planospiral form that kind of, goes out in this football shape as they spiral around their proloculus. And as they spiral, they add a chamber at a time, moving their antitheca one chamber forward. Each time it adds a chamber, and each time it does so, this causes furrows to form, aka the grooves left over from the previous antitheca after each chamber was added. So these furrows can be seen in these lines here in the outer part of the foram. And if you cut the internal part, you can't see the furrows. It's just on the outer part that you see the furrows. And then the septa are what separate each chamber. So similar to what we talked about earlier with sutures, it's just that the septa separate each chamber. And so we can see these septa are shown here. After each chamber, there's this little tick. Those are septa. We can see the axial septula shown here. Septula are just littler septa that don't reach all the way to the bottom, whereas the septa are the complete separations of each chamber that do reach to the bottom and do separate each chamber. We also have something called coma, which are kind of secondary deposits, or these dark lumpy things shown here. And as you can see, there's this like X looking shape of coma shown in the axial section. And this X shape of coma surrounds something called the tunnel, this part in the middle that doesn't have any secondary deposits or doesn't have any coma or comata, plural. That is called the tunnel, that inner part there, that lighter part. And then the X surrounding it is the comata or coma singular. And we also have some terms over here like karyotheca, or over here like spirotheca. Well, we'll talk about that as we get into the wall structures. Wall structures structures of Fusulinids can get a little complex, and I had to draw these so many times for my paleontology class, so I'm going to go into them now. Basically, the spirotheca is the spiral outer wall, which actually is formed in several layers. And we'll talk about what these layers are in just a second. First, I got to tell you what's going on down here. What does it all mean? Now, what these three different structures represent is kind of the evolutionary trend of Fusulinids. First, they had profusulinellid wall structures, a very ancestral simple wall structure shown in A. Then we had B with the fusulinellid wall structure, still fairly simple, but there's an extra layer we'll talk about. Then we had C, the schwagerinid wall structure, and these were a little bit more simple in some ways and a little bit more complex in others. So we'll talk about all three of these wall structures. Basically, these several layers that make up the wall structures include the tectum, which is the first organic rich layer, this dark line labeled T, and that's also labeled in the fusulinellid wall structure and the schwagerinid wall structure, and those are just the first organic rich layer, the tectum, and then inside this layer there's a thick calcareous layer, or the diaphanotheca. This is relatively thick, so we can see that's much thicker than the tectum layer. That diaphanotheca is calcareous instead of organic, and that's shown in both of the first two wall structures, the profusulinellid and the fusulinellid wall structures. And also in both of these wall structures, you have the chambers coated by a secondary organic rich calcareous material, so organic and calcareous material, and the tectorial deposits labeled by E, also sometimes called the epitheca, and this E label is shown up here in the profusulinellid wall structure and both above and below the wall structure in the fusulinellid wall structure. That was kind of the trend in evolution from the profusulinellid to the fusulinellid, was the addition of an additional secondary tectorial deposit or epitheca below the wall structure. And so this tends to be a 4-layer structure, and the profusulinellid tends to be a 3-layer structure. However, as we get over to the schwagerinid wall structure, we see that there are only 2 labels, and they're lacking the diaphanotheca as well as the epitheca or tectorial deposits. So in terms of this wall structure, we have tectorial deposits absent. We also have a layer equivalent to the diaphanotheca that is thickened, and small pores of the tectum unite to form larger passages through the translucent layer, and that is now called the karyotheca. This karyotheca structure is this complex, now honeycomb structure that allows the schwagerinid wall structure to be stronger. This is what we saw up here. This was the karyotheca of this schwagerinid wall structure shown here as well as down here, and they also have pores labeled here, and this wall structure was stronger. So the evolutionary trend from profusulinellid to fusulinellid to schwagerinid was going from relatively weaker to much stronger in their structure. So if that's the evolutionary trend for Fusulinids, that's great, Rachel, but what are the evolutionary trends among forams as a whole? We're here to learn about forams, not just Fusulinids, right? Okay, yeah, you're right. So let's move on to foram evolution as a whole. First, in the earliest of the Cambrian period, we see cylindrical agglutinated tube forams evolve, and then the foram evolution was a little bit boring throughout the early Paleozoic, in which their morphologies remained relatively simple, and then enrolled tubular forms appeared in the Silurian, and then we had multi-chambered agglutinated forms appear in the Devonian, and then by the Carboniferous, which is where this figure starts. This figure over here shows from the Pennsylvanian Permian time to the Triassic, Jurassic, Cretaceous, Paleocene, Neogene, and Holocene, so from older to younger going down, and also shows ecology of the foram going from nearshore to offshore, left to right. And by the Carboniferous, we see biserial, triserial, and trochospiral forms, as we see up here in the Pennsylvanian Permian row. Then we also note down here again, Fusulinids were probably the most noteworthy late Paleozoic forams just because of their really useful index fossil use, and so we see these up here as well. But during the Mesozoic, after Fusulinids had died, miliolines and lagonines or lagonines were allowed to radiate and dominate during this time, and then planktonic forams actually first evolved in the Middle Jurassic. They weren't even around in the entire Paleozoic. It was from the Cambrian to the Jurassic that benthic forams were the only things around. Then planktonic forams evolved in the Middle Jurassic, and then you had planktonic forams hit their peak diversity really soon after that in the Cretaceous. And then planktonic forms were hit really hard in the KT extinction. Only 3 species survived, but these species were able to recover and diversify in the early Cenozoic, and now all of the planktonic forams that we see today have evolved from those initial 3 species that survived the KT extinction. And then the benthic forams were hit hard in the Paleocene-Eocene extinction event during the PETM, which is the Paleocene-Eocene thermal maximum, a very warm period that likely melted methane hydrates, altering the bottom water composition of the ocean heavily enough to kill a lot of these forams. And then they suffered another extinction event at the Eocene-Oligocene boundary when things started to cool off, because nothing likes rapid change, whether it be warming or cooling. And then after this extinction event, you had planktonic forams recover and diversify in the early Miocene when things began to warm up again, but then the cool conditions in the middle Miocene led to further extinctions, and this remarkable dying and recovering and sensitivity to temperature makes these fossils great paleothermometers, or indicators of ancient ocean conditions throughout Earth's history, like we talked about previously. And in terms of evolutionary trends, some really cool trends were seen, especially in planktonic forams. For example, we see iterative evolution, or repeated evolution, observed in planktonic forams throughout the fossil record ever since they evolved in the Jurassic. What this means is that we see after each major extinction event diminished their morphological diversity, those morphologies that went extinct eventually evolved in new forms, which are called homeomorphs, when one morphotype or morphology of an organism goes extinct but then kind of evolves again in a new form later on in a convergent evolutionary event, because that morphology is beneficial for a certain environmental niche. That new form is called a homeomorph, and those homeomorphs formed every time these morphologies went extinct from the ancestral Globigerine morphotype. They just repeated this pattern after every single major extinction event took them all out. They kept coming back from this simple ancestral type of morphology of planktonic foram. But that is all I have for you guys today. So we have the upcoming video of the diatoms, radiolarians, and coccolithophores. And if you are still watching this video, before you go, why don't you comment down below for me what your favorite foram is? Mine personally is the genus Elphidium. I think they're really cool and have a really cool, intricate like ribbing on their test. Anyway, comment that down below. I'm really curious to learn your favorite or least favorite. If these have caused you much pain in a class or a test, I understand. Trust me, Fusulinids have scarred me. And I want to give a huge shout out to my TA when I took this class, Jenny Rishal, who is now Dr. Jenny Rishal and studies these things. Like, my God, you are such an inspiration. And she gave me a lot of the knowledge that I used to make this video. So shout out to her. And with that, thanks so much for watching, and I will see you guys next time. Bye. I forgot where I was. That is a problem. Sarcomastigophora. I said that so good, so fast, and I just ruined it by gasping. Granuloreticulosa? I said that good too. Oh my gosh. Is it sessile or sessile?
Sessile.
Oh, come on. It's sessile in the US and sessile in the UK. I want to sound like I'm from the UK. Where was I? I don't remember. Oh, fudge. Yeah, I don't have a Foram shirt, but I'm working on getting that, so excuse the stars.